Good afternoon, and welcome to the 2021 Project Lead Advanced Topics session. Thanks for joining us. I'm excited that we finally have some people here in person and a lot more of you out still in the virtual world. But we're excited to bring you this timely presentation on mRNA vaccines, a topic that potentially gives us a completely different approach to addressing breast cancer, both in treatment and prevention. My name is Tracy Solak, and I'm a 15-year breast cancer survivor. I'm a graduate of NBCC's Project Lead and Advanced Project Lead, and I'm also a YSC RISE Legacy Advocate. I've participated as a consumer reviewer in numerous DoD grant review panels, and I'm an advocate member of NBCC's Artemis Project, which you'll hear a little bit about more in a few minutes. Many of you already know about NBCC, but I want to give you just a little bit of background. In 1991, a small group of women invited breast cancer organizations from all across the country to create a coalition of those organizations. This separate coalition would bring these diverse organizations together to address overarching challenges in breast cancer through political action with a single voice and targeted goals. Its mission was not to replace each of these member organizations' missions and goals. Instead, it was to bring the organizations together to work toward larger goals. Since NBCC's inception, its mission remains unchanged: to end breast cancer. Our goals to achieve this mission have been to make sure there is sufficient funding for meaningful research and the right clinical trials, to ensure access to quality healthcare for all, and to advance the influence of trained advocates in all aspects of breast cancer. Our mission-driven strategy created a unique approach to breast cancer advocacy. As many of you know, NBCC's work has increased federal funding for breast cancer research through the DoD Breast Cancer Research Program, and advocates are involved in helping to monitor how all of those research funds are spent. Our activism in that area has generated more than $3.9 billion, new dollars, for breast cancer research. And our research initiatives and advocacy have helped bring about new models of research. Our vision and advocacy led to the first ever system of access to care for thousands of low-income women with breast and cervical cancer. And we've trained thousands of advocates who challenge the status quo and demand more wherever breast cancer decisions are made, from hospital boardrooms to Capitol Hill to research laboratories. One key component of NBCC's research work is the Artemis Project. Originally, it was created as the research component of Breast Cancer Deadline 2020, but the Artemis Project is an advocate-led, innovative, mission-driven approach involving strategic summits, catalytic workshops, research action plans, and collaborative efforts of various stakeholders. Although NBCC could not possibly fund the scope of research needed to end breast cancer, we could at least model what a mission-focused and truly collaborative breast cancer research program looked like, and to do it in the context of two overarching questions that are very important to us, but that were not receiving sufficient attention. We named the project after Artemis, the fierce Greek goddess of hunting, because who better to take on such an audacious project? We recruited talented and innovative researchers, advocates, and other key stakeholders from fields as wide-ranging as immunology, medical anthropology, biophysics, genetics, molecular biology, biomedical engineering, informatics, and clinical oncology. The Artemis Project focuses on two areas: primary prevention, how do we stop people from getting breast cancer, and prevention of metastasis, how do we stop people from dying of breast cancer. Artemis Project participants design and implement research plans and interact through an infrastructure maintained by NBCC that allows collaborations to thrive and progress rapidly. The next Artemis meeting will be in California this coming March. So we started the Artemis Project with the bold idea of a preventative vaccine, and many in science, the media, and other advocate groups looked on with mild and mostly dismissive amusement, but we kept at it. It's taken 10 years, but we are now preparing for a phase 1 safety trial of a preventative vaccine in partnership with the NCI Prevent program and the Mayo Clinic, led by Dr. Keith Knutson's lab. And NBCC has filed a provisional patent on the vaccine. At this stage, we're working with NCI Prevent to facilitate the manufacturing of the vaccine for use in a phase 1 clinical trial, although COVID has delayed this a little bit. Our ultimate goal is to develop a safe and cost-effective vaccine that targets all major subsets of breast cancer, reduces the incidence of breast cancer, and prevents death from breast cancer. And as you'll hear today, we're not alone in this work. Others are exploring how to harness vaccines for both prevention and treatment of breast cancer. Before I introduce Dr. Wang, I just want to mention 2 other educational opportunities that are available through NBCC. The first is the Project Lead Institute, which will have 2 sessions next summer in La Jolla, California. Project Lead is a week-long deep dive into breast cancer science, training advocates in what they need to know to be able to make their research advocacy more effective, both on the science side and the public policy side. The dates and applications are on this slide, and I can provide you with more information afterwards if you need it. Also, the Advocate Leadership Summit will be held in Washington, D.C., April 30th to May 2nd. There's plenary sessions and workshops on a variety of topics related to both research and public policy, and ample opportunities to network. And then that is followed on on May 3rd by NBCC's annual Lobby Day, where we go and meet with senators and congressmen to advocate for both legislative and public policy priorities related to breast cancer and NBCC's mission goals. Again, with that, if you have any questions, feel free to come see me afterwards. And now for our topic today, mRNA vaccines. These have been in the news for the past year, obviously, because of COVID-19. But what many don't realize is that this type of vaccine has been studied and developed over many years, and they may have a variety of applications, including the potential for cancer treatment. Dr. Wang will explain how this might become a new paradigm in breast cancer treatment and for all breast cancer patients. So I'm pleased to introduce Dr. Nathaniel Wang. Dr. Wang is the Chief Executive Officer and Chief Science Officer at Replicate Bioscience and a co-founder of the company. He has over 15 years of leadership experience in immunology and drug development. His work has focused on the area of synthetic self-replicating RNA technologies and their delivery, including the development of the Synergy technology platform. He is also a recognized expert in the fields of immuno-oncology and infectious disease. And today, Dr. Wang will be talking to us about mRNA vaccines, how they work, and what possible applications they may have in breast cancer. And I'll now turn things over to Dr. Wang.
Hi, Tracy. Thanks so much for that kind introduction. And I'm excited to be able to speak to everybody today. I've been hoping to be there in person, but unfortunately my flights had gotten canceled. But I'm still excited to be able to speak to everybody virtually. And along those lines, since unfortunately I'm not there in person, if there are questions after the session or things you think of later, since I can't see all of you in person, please don't hesitate to reach out. I'm sure the coordinators can provide my contact information. I'd be glad to speak to anybody separate from this as well to answer any questions that you would have. So after that kind introduction, I'd like to start and tell the audience a little bit about RNA-based vaccines and how they're being applied in the breast cancer space. So I'll talk a little bit about some of those clinical successes that Tracy was referencing, especially in the COVID-19 space, talk a little bit about self-replicating RNAs themselves, how they can be developed to target drug resistance in oncology, and specifically breast cancer, and then give a case study of a clinical trial that had been recently run in the space that shows how the technology can really work in terms of vaccines. So for RNA vaccines in general, I think it's good to contextualize it for most of history and certainly well into the 20th and 21st century. It usually took decades and sometimes up to 100 years to develop a vaccine. And I think what we've really seen with a lot of these synthetic technologies, particularly RNA, is how rapidly you can develop different types of countermeasures with these new unconventional types of technology, where we were able to essentially go from a pathogen sequence to a vaccine in the course of about a year or even a little less than that, including all the stages of clinical testing. So, just to talk about the difference between mRNA and self replicating RNA in general, the way that I like to think about mRNA is an instruction manual that your cell uses. And imagine that proteins are a bunch of different Legos inside of the cell, and it needs this instruction manual to tell it how to build different types of proteins. I personally have a couple of toddlers at home, and so I'm very familiar to how they ruin anything, especially when it's related to things like instruction manuals. Um, the way that I would liken messenger RNA is that that instruction manual will degrade after about 24 to 48 hours. Imagine things being spilled on it all the time within the cell. With self replicating RNA, what you're essentially doing is delivering a copy machine into the cell along with that instruction manual. And so the cell is able to spin out a lot of copies of that instruction manual. And so you end up generating a lot more protein for every copy of that original instruction manual that you've delivered. And this can lead to functionally lower doses or better immune responses in the context of vaccines. So, similar to other types of synthetic technologies, RNA vaccines, including self replicating RNAs, are designed really for patient friendly administration. So, they would be handled with a needle and syringe and the deltoid, just a half mil dose. The current storage is either at 80, minus 80 Arctic type temperatures or minus 20, and the field is working actively to get it to a little bit more stable forms of lyophilized or freeze-dried versions of the vaccines. And all of these are amenable to either single dose or repeat dosing. And so it's a very versatile technology from that point if you need multiple doses of something. So again, I think that we've all seen a lot of recent successes with COVID-19 mRNA vaccines have really shown the promise of the approach. And so there are three major buckets and ways to think about different types of mRNA. There's mRNA where the individual components and the individual bases of it are unmodified. And that's something similar to what CureVac had used in their trial. And while that had shown some efficacy because of the low doses that they administered it at, it was unfortunately more around 50% efficacy. There's the base modified mRNA, which is better at hiding from the immune system and preventing some of that systemic non-specific inflammation that leads to some of those toxicities that I think many of us have experienced with the current vaccines from Moderna and Pfizer. And those vaccines are, of course, approved and are very efficacious. And then you have self replicating RNA. The technology is still fairly new and it wasn't quite ready uh, when the pandemic had hit, but there were certainly some trials from Imperial College London with phase 1 trials that have completed, and Arcturus Therapeutics is currently in phase 3 trials with a version of that technology as well. So it's certainly shown itself to be very safe and well tolerated in human populations, similar to the other types of mRNA technology. And so it's really building off of these successes and the infrastructure that was developed as part of COVID-19 to really accelerate the development of other RNA based therapeutics, especially in the areas of oncology, such as in breast cancer. So, just a little bit about how RNA vaccines are made and some of the benefits that come from that. So, RNA technology is a completely synthetic process. So, essentially, you have some kind of steel bioreactor. This can be, anywhere from a liter to 1,000 liter bioreactor, and you produce the RNA itself from DNA templates using enzymes. Uh, that material is then purified, and some of those DNA fragments and the enzymes are removed from it. And there's a filtration step. And that's the active RNA that would then be ready for formulation into the drug product itself. So you get highly pure material coming off of this type of product. It's very rapid, taking less than 8 hours, and it can be very high yield. The point being that you can get a large number of human doses from a very small reaction, which is important for accessibility to a lot of different populations, which is something that we believe passionately in. So once you have that active RNA molecule, you then formulate it into a fat bubble called the lipid nanoparticle that's then used for delivery and is suitable for injection into a person. And so there are four different lipids that comprise that lipid particle. You combine that using different types of mixing technologies with that RNA payload, and it spontaneously forms these particles that you can then filter and isolate, and then those are what would be ready for injection into a patient themselves. And as Tracy had alluded to, while RNA vaccines have really recently come into the public consciousness because of COVID-19, this technology has been developed essentially since the 60s and the 70s. The first vaccines of these types against infectious disease were really shown in the late 80s and the 90s for the delivery of different genes and for vaccines such as flu for influenza. It was really in the early 2000s that this four component fat bubble was developed in ways of really scaling up the manufacturing to develop it from global manufacturing perspective. And then, of course, all of the development in the last 10 years that really culminated in the global COVID-19 vaccine. So I just wanted to talk a little bit about how self-replicating RNAs are practically differentiated from mRNA. And so if you look at mRNA today, again, like from what you would see in the Moderna or the Pfizer vaccine, that's the gold standard. And so we would set that to a value of 1. I referenced that both Imperial College London and Arcturus Therapeutics had been running clinical trials in the space for COVID-19. And if you look at the effective doses that they were using on patients, it was 6 to 20-fold less than conventional mRNA, depending on the exact comparison that you were using. And we think that the doses can even be substantially dropped further from there to 1,000-fold less with the current iterations of the technology and perhaps even further in the future. And I think the importance of that to patients is that you would have far less side effects and you're more likely to be able to generate very robust immune responses, even in challenging settings like cancer, uh, where you certainly don't want to add to the side effects that patients are already feeling from the current types of treatments that exist. This will also functionally lower the cost of goods for patients, which I think is something that's incredibly important, that we don't just focus on developing designer therapeutics that are accessible to a lot of patients but can be expensive, but we also develop these types of cost-effective interventions as well. So in general, for self-replicating RNA, they've certainly been very well tolerated and performed well in humans across a variety of different studies in both infectious disease and oncology. So this isn't a new technology. It's been in a variety of different phase 1, 2, and even phase 3 studies. So in infectious disease, it's been in humans from anything from influenza to HIV, to, of course, SARS-CoV-2. And on the oncology front has been tested anywhere from CEA, for colorectal cancer, PSMA for prostate, HER2 for, of course, breast cancer, and a variety of other indications as well. And so we'll be speaking today about one of the case studies that related to HER2 that some of our colleagues at Duke University had run. So this is just to say that one of the ways that we achieve those lower doses is by optimizing all aspects of that drug product that we had shown you previously. So how it's actually delivered in that fat bubble, the specific type of vector to make sure that it's something that's safe and can drive those lower doses for patients, and exactly how you design the different types of protein that go into the different candidates. And importantly, working very hard to remove any of the contaminants that can arise in manufacturing that can lead to some of that reactogenicity that many of us are familiar with different iterations of the current RNA vaccines to further improve on the process. So this next section will talk about the specific development of vaccines that can target drug resistance and cancer. So, something that I think a lot of us are familiar with is a concept that many of deaths related to cancer actually come from drugs uh, that fail the patients ultimately. And I think that we also ultimately understand, and I think this is a little bit more intuitive, that treating cancers in earlier stages is associated with better outcomes. And by earlier stages, we don't just mean earlier stages of the tumor itself, which is also important, but also earlier stages of care. So coming up with improved first line treatments rather than just subsequent line treatments. And all of that together have led to a real unmet need for safer, effective treatments at those earlier stages of care. So I think it's important to shift our focus as we go forward in the future to trying to, as Tracy had mentioned in the Artemis Project, first look to prevent cancer from occurring in the first place. And if it does occur, to help prevent some of those disease transitions as early as we can catch it, which is something that would be beneficial to all patients around. So that the way that we would do this is, so anytime there's a tumor and you treat it with some kind of targeted therapy, something like a small molecule or some kind of antibody, often there's an initiating event where an individual cancer cell either has a pre-existing mutation or will develop a mutation against whatever that therapy or drug is. As there's that positive selection pressure on that individual cancer cell, it will continue and be selected for expansion within the tumor itself, eventually presenting itself as clinical resistance and eventually causing the drugs to fail patients themselves towards the end of that as it starts to overtake the rest of the tumor itself and spread that resistance. And so, what we're really focused on doing is catching these initiating events as they occur in real time and remove those cancer cells before they can spread to the rest of the tumor and spread that resistance. And so, one of the ways that we're doing that is using this concept that we're calling synthetic immune lethality, which we think is a novel approach to really bridge between those small molecule or antibody treatments that patients are already receiving and some of the newer types of immune therapies. And so, the way this works is the following. So, if there's no treatment on the tumor cell itself, it will continue to grow. If you administer some kind of targeted therapy, think tamoxifen or aromatase inhibitors in the case of ER-positive breast cancer, oftentimes the estrogen receptor itself will mutate in certain residues that confer resistance to those different types of small molecules that will lead the tumor cell to escape and continue to grow. If you just administer a non-specific immune therapy, or you educated the immune therapy to recognize some of those resistance mutations, if there's not that selective pressure caused by those standard of care small molecules, then again, the tumor cell can simply not mutate those residues and continue to grow. And so, what we really do is are proposing that while patients are on those standard of care small molecules, that we would concurrently administer one of these RNAs to educate the immune system to recognize those resistance mutations that arise in response to those therapies. And so, it's a lose lose situation for the tumor cell. If it does not mutate in response to the current standard of care treatment, it will continue to be susceptible to that treatment. If it does mutate those individual residues to gain resistance to the small molecule, it will then be susceptible to our immune therapy approach. And so it's this forcible coupling of these mechanisms of action that really create this lose lose situation for the tumor cells. And so the way that we envision this working clinically is this would either resensitize patients who already have these pre existing mutations or delay the onset of these resistance mutations that ultimately lead to those different drugs failing patients. And so, to continue the example of this in ER-positive breast cancer, there's a variety of different mutations that end up arising and that have been described in the clinical literature. And it can seem counterintuitive at first. There's actually a small number, but there's actually choke points in all of these different cancer pathways. So it isn't just an infinite number of mutations that end up arising. It ends up being a very finite and very specific number of mutations that occur. So there's two major types of mutations. There's on-target mutations, or mutations that occur in the direct target of those small molecules, like SERDs or aromatase inhibitors, and those would be activating mutations in the estrogen receptor. So mutations that turn the light switch on, that render it insensitive to those different types of drugs. There's also a different class of mutations called bypass mutations, which are turning on other pathways that render signaling through the estrogen receptor to be redundant or irrelevant. And so examples of that would be activating mutations or mutations that turn on PI3 kinase, or amplifications in genes such as HER2 or HER3. And so the net result of being able to target all of these different mutations is you would be able to create a treatment that would be accessible to all ER positive breast cancer patients, and a treatment that's off the shelf, something with a high therapeutic index that would be expected to have very low uh, adverse events or side effects for everybody, that could be combinable with the current standard of care, regardless of the line of treatment, and something that could eventually be pushed into earlier lines and earlier stages of the tumor itself. For instance, in the case of getting or receiving adjuvant therapy. And so this is the first step in something that hopefully strikes everybody here as very ambitious, and something that is designed to be for all breast cancer patients who are positive for the hormone receptor. So, as an example of this type of approach, if you look at encoding mutations that activate the estrogen receptor in the context of a viral vector, in this case, an adenovirus, and this was done with our collaborators at Duke University, what you can see is for a tumor cell line that was implanted into mice that expresses this mutation that renders it insensitive to different types of hormone therapy, you can see that this vaccination approach is able to control the tumor. And the way that it controls the tumor, shown on the right here, is by eliminating that particular mutation that is rendering it more active, the estrogen receptor itself more active. So this is good proof of concept for targeting some of those on-target mutations that I had mentioned on the last slide. And of course, this is all combinable with targeting those bypass resistance mutations. And so for one of the candidates that we're developing at Replicate, again, what we're doing is encoding all of these different resistance mutations into the same candidate, so that, again, you're capturing the vast majority of ER positive breast cancer patients who could receive this. So again, mutations in PI3 kinase and the estrogen receptor, as well as targeting amplifications in HER2 and HER3. And what we're showing on the right-hand data piece of this slide is just when you administer this in mice, you're able to generate robust immune responses, and importantly, robust T cell responses against all of these different mutations. So there's over 6 mutations in the estrogen receptor, 4 mutations in PI3 kinase, again, as well as uh, amplifications in HER2 and HER3. So conceptually, this shows the benefit of this type of approach. And these are currently in efficacy studies, and we're looking to push this candidate clinical in the next 18 to 24 months, if not sooner than that. So shifting gears a bit for the last part of the talk, just wanted to give a case study that talks about a HER2 vaccine that was based on the same type of fundamental technology of self-replicating RNA, and what that looked like in the clinic. And so in this particular case, this is a particle version of this technology. So instead of having a fat bubble that it's delivered in, it has a viral shell that it's delivered in, but it's ultimately based on the exact type of viral family as the other types of self-replicating RNA that we were speaking about before. And that's specifically alpha virus self-replicating RNA. And so in this particular trial, I was looking at stage 4 metastatic breast cancer patients who are progressing on HER2 antibody therapy. And they were administered 3 different doses of this self-replicating RNA that was encoding this truncated version of HER2, the extracellular domain and the transmembrane domain, along with continuing to receive anti-HER2 standard of care therapy. A, a key part of this, and in line with everything that we've been speaking about during the course of this talk, is it was very safe and well tolerated, either alone or in combination with standard of care. And while there were grade 3 adverse events, those adverse events were not attributable to the vaccine portion of this regimen. So, that's very positive in showing the benefits of using these types of technology and the high therapeutic index that these have and how they can be combinable with different types of standard of care. The other aspect of this is using unbiased clustering analysis, which is a fancy term of being able to assess blood responses and immune responses in the blood to different types of targets that are induced by the vaccine, and just showing that you can generate strong CD8 T cell responses. And those are the killer T cells that are responsible for destroying the tumor cells themselves. Perforin itself is just the name of a protein that's directly involved in the ability of T cells to lyse tumor cells, and just showing that these types of genes and responses were upregulated following vaccinations and were a good biomarker of response to this vaccine. So in the phase one trial in patients who demonstrated this biomarker of immune responses had a positive effect on their progression-free survival, even in a challenging setting, and suggestive improvements in overall survival. Interestingly enough, this also tracks with some other studies using an identical version of the technology, but encoding CEA that was tested in colorectal cancer patients, where you saw similar breaking of tolerance and a strong amount of T cell responses in terms of clinical responses. So again, this is showing that conceptually, you can generate very meaningful T cells in patients that are suggestive of being able to help protect against different types of tumors. And so this is something that is certainly currently in phase two testing right now at Duke in combination with checkpoint inhibitors. But as a specific case report, looking at the combination of the vaccine encoding HER2 along with anti PD-1 therapy, this was a 62-year-old woman who had metastatic HER2 positive hormone receptor negative breast cancer who had been progressing on anti HER2 therapy. In this case, had received 4 doses of the vaccine along with checkpoint inhibition. And what you saw was an increase in both helper T cells, which help the immune system recognize other types of proteins in the tumor that you are not targeting, as well as ones that you are. So it helps spread and broaden the immune response, as well as some of those killer T cell responses that can directly kill the tumor cells themselves. And what the team was able to see after a single cell analysis was that there was no detectable tumor cells by that analysis post treatment. So, again, while this is a single case, I think it is um, certainly encouraging at the types of responses that you can get uh, taking these types of vaccination approach and combining them with other types of agents clinically. And so, again, just wanted to take a step back on everything that we've been talking about. And again, really focus in on this concept of disease interception and preventing some of these transition events that lead to disease. And so, while we were discussing, especially about our own internal programs that are focusing on preventing the transition from a non resistant to resistant state later on, I think what we ultimately would like to drive to as the field moves forward is to prevent the transition point really between occult disease and clinical disease very early on and intercept the disease before it ever takes off. And I think that that's something that is really achievable going forward, especially as we increase the amount of biomarkers we have and become better at sampling patients' blood early on and catching these events early on. I think we'll be able to develop different types of medical interventions, such as these vaccine approaches, to really shore up the immune system and educate it to recognize these to prevent that. And again, I think that's incredibly important because I think the current way that we're approaching a lot of the oncology space is in later stages of disease where we develop agents that are going to be too difficult for patients and will not do a good enough job of serving patients earlier on in the disease itself. So that would be our vision in the space. So again, as some key takeaways of targeting endocrine resistance in ER positive, HER2 negative breast cancers, self-replicating RNAs in general are well tolerated and can generate robust effector memory T cell responses in humans. Most endocrine therapy resistance is caused by mutations or amplifications in the estrogen receptor, ESR1, PI3 kinase, or amplifications in HER2 and HER3. And so, our approach is by targeting all of these different types of mechanisms of resistance that cancers are able to develop to help make them more visible to the immune system. So, one of the ways that you can imagine this by analogy is if there's a wanted poster, and every time a tumor ends up developing a resistance mutations, imagine putting on a mustache or putting on a pair of glasses or putting on uh, different ways of changing what that wanted poster looks like. And what we're trying to do is remove all of those mechanisms that the tumor cell and the tumor itself uses to disguise that wanted poster. And that way, we're able to find the real culprits that are driving a lot of that disease. And then finally, anti PD-1 therapy or checkpoint inhibition can synergize with vaccines, both in preclinical models, although we haven't shown you that here, as well as having suggestive improvement clinically. So with that, I wanted to thank you all for the time and leave plenty of time for any questions that people have. And so I'll stop there in terms of showing the slides and are happy to tackle any questions people have.
Thank you, Dr. Wang. I do have one. This is Tracy to start off. You've talked a lot about ER positive and HER2 positive. How do you envision that this technology or this type of vaccine might work for someone with triple negative breast cancer?
Yeah, absolutely. So a lot of the types of pathways that we are targeting, things like PI3 kinase activating mutations or HER3, are certainly implicated in triple negative breast cancer. And obviously, HER2 is implicated in HER2 positive breast cancers. And so, with this type of candidate, while the initial focus would be in ER positive breast cancer, the hope is to expand it into those other indications as well, including triple negative breast cancer eventually, as well as HER2 positive breast cancers. So it is something where we're building it with that in mind to be able to expand to all of those other types of breast cancer populations, which is critical for us. The other aspect is as we learn more about these different types of mechanisms, of resistance, we'll be able to encode those types of genes into follow-on versions of this product. And so, again, one of the benefits of RNA technology is the time it takes from discovery of those new types of resistance mechanisms to create an actual candidate that incorporates them is a very short turnaround time once you start generating some early clinical data with this type of approach. And so that's where you see where RNA can really be transformational in really condensing and accelerating the development of these types of therapeutics, even in the oncology space.
Thank you. Are there questions from the people who are live? Bob, Bob is coming, the second tallest advocate in the room.
Hi, thank you very much. When I think about cancer vaccines, I think about something like Gardasil, which is sort of an antivirus type thing. And here you're talking about vaccines. I mean, I don't think of breast cancer necessarily being a virus per se. So, can you maybe just sort of clear up this mud in my head about are vax? I guess vaccines do more
Yeah.
than stop viruses.
Yeah, absolutely. That's a fantastic question. So the way that we think about it is what vaccines ultimately do is educate the immune system to recognize again that wanted poster. And sometimes that wanted poster is a pathogen or a viral protein. In other cases, it's versions of our proteins that have been mutated. And in both cases, both of those are foreign to us, right? Our protein should not be activated like that all the time to cause cancer. And certainly a pathogen protein is also something that's foreign. And so we're training the immune system to recognize things that are foreign. And so what's similar to this type of approach to the infectious disease approach is similar to how Gardasil is teaching you to recognize a virus before you actually get infected with the virus. we're teaching the body how to recognize these mutated proteins before the body encounters these mutated proteins. And that's why it's fundamentally a very different approach, even in the cancer vaccine space.
We have a couple of questions from the people who are watching virtually. The first one, are vaccines being studied in patients who are already metastatic, or are you looking at it only in the context of prevention?
Yeah, absolutely. So they're being looked at in all different types of populations. So they're being looked at metastatic breast cancer patients, oftentimes as part of subsequent line therapy. We're starting to look at it at earlier stages of care. So we want to look at it in first as part of first line therapy. And then some in the field are also looking at it at earlier stages of the tumor itself. So there's certainly been some recent data, for instance, in stage 3 type tumors, both in colorectal cancer, but also in breast cancer, that have had promising results. And I think one of the most difficult aspects of those trials is because the follow up is so long, which is a great thing, and we should all be grateful for that. It does make for a more challenging trial in terms of how long it takes to get the data readout. And so I really think that's where having good partnerships with organizations such as Artemis or with the US government or with other types of commercial organizations, and really everybody working together to take on those difficult types of trials, is something that's in everybody's interests.
So another question from the virtual community, does I guess in the current form of what you're working on, is there a way for it to address P53 mutations?
Yeah, absolutely. So P53 mutations are definitely something that you can encode into these different candidates. What's interesting about P53 is it's not implicated as much in terms of a resistance mechanism. And so that's one of the reasons we don't target it, although there are other candidates out there that do target it. Examples of RNA candidates where they are targeting P53 would be current candidates in the colorectal space and in the lung cancer space from Gritstone Bio. One of the challenging parts about P53 is sometimes the expression of P53 can block the responses to the other genes that you're actually expressing in that same cell. And so, from that standpoint, it can be difficult to combine P53 with some of these other mutations. But short answer, yes, people are targeting it. Yes, we could target it as well. No, it's not part of this first generation of it, but something that we're considering for the subsequent versions of it.
There's a question about the uh, study that you had talked about a little bit. How long has that patient who was HER2 with no evidence of tumor survived, and how long has she continued, assuming it's a she, to show no evidence of disease?
Yeah, absolutely. That's a great question. And, I don't have the most up to date information on that. Again, that was generated with our colleagues at Duke. That's certainly something where, if there's further interest in that question, I'd love to connect uh, directly to that team on that.
And would liquid biopsy play any role in detecting the tumor in time to allow this vaccine to maybe work earlier and prevent a transition from an early stage to a later stage?
Yeah, absolutely. So, with the genomics revolution and the increase in a lot of those different types of technologies, what we've essentially been finding across all types of cancers is that you can find these early transformation events in cells in almost every tissue that we end up sampling. And there are certain tissues that are more prone to it, tissues like breast cancer or breast tissues or lung tissues or other types of mucosal tissues that are particularly susceptible, and that's probably because they interact with the outer environment a bit more. And the idea is absolutely, as we start to detect some of these with greater and greater sensitivity, that we would be able to administer treatments to patients, such as vaccines, that would really start to function more and more like that Gardasil or other types of vaccines that other people have referenced, where they could be given when a patient is essentially completely healthy and really delay the onset of disease if not prevented entirely from occurring in the future. So that's absolutely an area of very active interest that we think that the improvements in genomics is really driving. So whoever asked that question really hit the nail on the head for where we see the future of these types of technologies.
So I think we touched on this a little bit before, but is the goal to be both, to be an early treatment and or an early prevention and perhaps a later treatment, or are you focusing more on one than the other?
I think that and we believe it's incredibly important to be addressing both. From a practical standpoint, in terms of showing that an approach is working the most rapidly, I think uh, that's why we are focused currently on later stages of the tumor, although again, earlier lines of care within that. And then once we see success there, it really opens up avenues into a use expansion into a lot of different areas. And so we're trying to get answers to the field and to patients as quickly as possible. And once we have some of those, we'll be able to answer with better data in hand exactly where we can expand that into. So uh, you need to learn how to fly before uh, you build a rocket ship.
Um, and then a question that I think goes to sort of a definition of prevention. If you're envisioning this as a tool for prevention, how would you select the population to receive it, since the majority of breast cancers aren't inherited?
Yeah, absolutely. And so what ultimately ends up happening is we envision selecting this with patients who are about to undergo some kind of endocrine or hormone therapy. So we know that that endocrine therapy itself is what's putting a selective pressure on the tumor to develop these mutations. And so we want to co-administer this vaccine along with the endocrine therapy so that the immune system can learn to recognize in real time those mutations that would lead uh, things such as endocrine therapies to uh, fail the patients. And so that's the current use case that we're thinking. I think that answers your question on how we're doing the selection, but as part of that first line therapy.
Okay, so it's more of a first line therapy than like a population based prevention like Gardasil. Is that more where this is going?
Again, so for the first type of clinical trial, yes, that's where it's going. Where we would like it to go in the future is more population based prevention. So again, we are trying to get clinical answers as quickly as possible. And with positive results there, we would absolutely want to push it into a more population-based prevention. But again, I think that there's a lot of complex questions that require advocates and policy and regulatory input because it's hard to really pinpoint what you would call efficacy in those population-wide assessments where you're trying to prevent something.
So, for those of us who just recently got a COVID booster, is this approach something that you would envision needing boosters? And if so, how frequently?
Yeah, that's an interesting question. So, one of the issues with current versions of mRNA technology, like in the uh, Pfizer or the Moderna vaccines, is they don't generate very durable immune responses. And one of the reasons for that is uh, because they look so much like self, because RNA can look too much like what we normally have in our cells, it doesn't stimulate a strong enough immune response. And so that immune response starts to wane or die down after about 3 to 6 months, which is why we need different types of booster treatments. With these vectors that are still fully synthetic and RNA based, but had their origination in viruses, those typically generate immune responses that last for much longer. So oftentimes they can last for, well over a year, including in the memory types of responses that they generate and the T cell responses. Eventually, might a patient need a booster? Probably, but we wouldn't envision after the initial regimen that they would need something on the order of every 6 months. We imagine that it would be longer than that. But again, it depends. Are we talking patients who are in very early stages, in which case you might get one every few years? Or are we talking about, later stage patients where they may require it something like every 6 months or every year? So it just depends on the use case.
So, as a layperson, this sounds very expensive. It sounds like something where the tumor's going to have to be evaluated to formulate the vaccine to look for specific mutations. How is this a cost-effective treatment?
Yeah, absolutely. So the cost of a dose of something like this at scale would be about $1 a dose. Right. So even if we have to administer 5 vaccinations to a patient or something like that, you're talking about the course of treatment costing about $5. In terms of being able to assess whether patients have these types of mutations, uh, acquired resistance mutations are used widely in clinical practice since they have predictive and prognostic value, and to help determine what subsequent line care is going to be. And so these are already diagnostics that exist today that clinicians often use. And so we aren't proposing using anything that isn't already currently in use from that standpoint. And the treatment itself, again, has very low cost of goods. And that's part of our passion and our goal of making these types of things very accessible.
So, could one vaccine target multiple mutations?
Absolutely. So, our current vaccine candidate targets at least 12 different mutations.
Are there any others from the room? I'm reading lots of virtual questions.
Uh, I have a question, and it may not be relevant, but I have been really curious as to the median in which these particular proteins are administered to the person. It showed originally that you would have it would be injected in the upper arm, I think, if one of the original slides. What medians do you use with all of these proteins when you do administer it to somebody? Am I making myself clear? I'm not really sure because I understand that all of this, it's all filtered and then it's clean and you try to get everything out. I'm just curious, what does that actually look like then, and how do you get it into a person?
It would look like, and it would be administered identically to, a COVID-19 vaccine. So you just have a little vial with some clear liquid in it. You draw it up in a needle and syringe, and you just inject it like you would just about any vaccine that we end up getting. So it would be indistinguishable from that standpoint.
Wondering what the clear liquid is.
Yeah. So the clear. Sorry, I'm hearing a bit of echo. That's why I was pausing. The clear liquid itself would usually be something like either phosphate buffered saline, so just salt water, or it would be something like a tris-based buffer. So tris is just a salt that's similar to uh, the phosphate that's used in regular salt water, and it has better buffering capacity, which in lay terms just means that it makes for a more stable product. It's something that you often find in most pharmaceutics. It's nothing strange. So it's basically a slightly different version of salt water. And in some cases, you might also have something like a sugar in there as well, just like a simple sugar that helps with stabilization during freezing. Something like sucrose.
Thank you. That answered. Thank you. That answered my question with regard to stabilization. So, thank you very much.
Of course.
Going back to the trial that you had talked about, are there, what is the stage of that trial? Are there trials open now for patients that are using this technology? And if so, is it just breast cancer, or are there other cancers where this technology is being used and there are trials available?
Yeah, absolutely. So there are certainly trials using RNA technology for triple negative breast cancer. that are currently open. There are trials in combination with checkpoint inhibitors for HER2 positive breast cancers, and there's multiple candidates there as well. There's other cancers like colorectal cancer. They're using RNA-based technologies. Lung cancer is another one. Pancreatic cancer is yet another one. There are also prostate cancer approaches. So there's a lot of different clinical trials using these types of RNA technologies. Uh, some of them are enrolling, all of them are at different stages.
So I saw on the one slide that it looked like there were not many adverse events from the vaccine itself. Are there challenges that arise in preventing unintended cross reactions with normal cells?
Yeah, absolutely. So I think that one of the benefits of a vaccine approach is you're expressing these proteins in a normal setting, so to speak, for your body to look at them. And so, for instance, it will naturally have feedback mechanisms where it won't target every cell that has HER2, just cells that are overexpressing HER2. And this is in contrast to things like CAR T therapies or other cell based therapies, where if they act too robustly in patients, they can cause a lot of those dangerous side effects where they attack tissues that they're not supposed to. And then also targeting some of these mutations don't occur in normal cells. They're by definition specific to tumor cells. And so any cell that they destroy would be a tumor cell. And so there are multiple layers of protection using these types of approaches that prevent some of those very scary kind of side effects you can get that can be life threatening using other types of technologies. It's not to say that those aren't always something that we worry about and test for and monitor for, though. It's very important to monitor to make sure there aren't autoimmune events that could have negative effects for the patient.
Certain types of metastatic disease present more of a challenge than others, particularly brain mets, and in some respects bone mets. So would this type of treatment be effective in those sort of hard to reach areas?
Yeah, I think one of the interesting thing about a lot of these acquired resistance mutations that arise in response to these treatments is that they can often uh, be found as circulating tumor cells or particularly enriched in certain types of metastases. And so this type of approach could work extremely well in that particular setting. So it's something that we think um, will have advantages in those metastatic settings versus other types of treatments.
I'm getting one last question in here. Have there been experiences in which the vaccines resulted in increased tumor growth, as can sometimes be seen in targeted immune therapy?
Yeah, absolutely. So, one of the things that often occurs is when you generate these robust immune responses, whether with a vaccine or some kind of other type of immune therapy, is you get a lot of cells from the immune system that go into the tumor itself, and the tumor appears to grow. And even as they're starting to kill some of those tumor cells, that's causing inflammation and swelling in the tumor itself, where it looks like it gets larger. So, one of the things that can be challenging for running these trials is looking at overall response rate or these other metrics that were really built for small molecules that directly lyse and shrink the tumor can be misleading. And so that's why in most of these vaccine based trials, in order to see an effect, you need to be either monitoring other types of biomarkers, like the presence of these mutations in the blood itself, or things like progression free survival and overall survival, of course. And so that can lengthen the amount of time that it takes to run one of these trials. But that's the practical outcome of the fact that sometimes these tumors can swell or grow larger in a way that's not actually something that's negative, but in something that's actually very positive.
Great. Well, thank you so much. We just have 30 seconds left, so I will thank you for being flexible with your ability to be here via virtual world instead of real world, and thank the people both here in the room and also out who are attending virtually um, for listening to this presentation. Thank you for your work in this field, and we will hope to see you at the next events.
Yeah, absolutely. Thanks to everybody for listening. I really appreciate it. And again, please reach out if you have any additional questions. We'd be glad to engage. It's a place we're passionate about.
Thank you.